So, can you hear the one? Yeah. So what I'm going to address you is about basically our work in the last 20 years, which is about the preconceptional space. This is the idea to walk you through that, and of course to get your questions later on and to get your input. So first, I would like to disclose that basically, as mentioned, I have no conflict of interest what I'm going to present. I was the founder of Igenomix, but now I'm not in Igenomix anymore. It was acquired by Vitrolife. It's a different thing. However, I remain as scientific advisor of Igenomix as well as other companies. I create my own non-profit foundation in order to continue our work on the research on women's reproductive health, trying to do as we have done in the past, from the bench to the bedside, and the example you are going to see this afternoon. And also we have created some startups coming from this idea devoted to specific topics on that. So this will be my disclosure. What I would like to address you during this afternoon basically is about the concept of precision medicine applied to the periconceptional space. Precision medicine is not, I mean, in oncology has been changed the way that oncology is being done. If you see in the induction of ovulation part of our field, precision medicine is what we used to do, trying to prepare the best treatment options according to the patient, according to the different parameters that we have. But when we just come to the end of the process, the most important one, which is when the embryo is going to attach to the mother, we still lack metrics and we still lack the precision that allow us to individualize the treatment of our patients. So this is one very important point that I want to make that will go through the whole presentation. And then specifically, I will address the first two parts that we have been going through and going from the bench to the bedside, which is the microbiome of the human endometrium, and later on receptivity, which is basically the initial step when the embryo adheres to the endometrium. So having said so, let me introduce you the periconceptional space, which is this. This is the black box that when we transfer the embryos to the maternal uterus, we do not know what is going on there. We just wait for 10 days to do a pregnancy test. But this black box is getting now unraveled, and we know actually what is happening there. And that's why I think it's important, because this is when the results really rely at the end of the day. We are able to get embryos. And if you see the statistics, we can get oocytes in many of the cycles. We can get embryos in most of the cycles. But once we transfer the embryo, then live birth is 40%. So we are missing here something. And I think this is the key element that we should really advance in order to improve the result for our patients. So this is some data that we have produced in my own group. These are the publications concerning to this. But of course, there are many groups worldwide working on these topics. And this is exactly what the embryo does when it goes to the endometrial cavity, either because go through the tubes or we transfer them. Once the embryo, the blastocyst, is there, the first thing that confronts is the luminal epithelium of the mother. And what is there is going to be the first thing that will affect this process. And this is why I will explain you what we have learned about the endometrium, the microbiota that is present there. Then there is a crosstalk between the mother and the embryo. There is a very nice crosstalk. The mother is passing molecules that modify the transcriptomics of the embryo. The embryo is talking to the mother at this time. And there are very nice work done about that. Nothing has been translated yet. So I will just focus on the next step, which is endometrial receptivity, which is at the moment that the embryo will be allowed to adhere to the endometrial, to the epithelial monolayer. And if it adheres, everything will start. If this monolayer is refractory, nothing is going to happen. So, and from that, it comes then the invasion part, which depends very much on the decidualization of the stroma, that has many things to do, how the pregnancy will go, and has a huge impact on preeclampsia, for instance, return birth, and this is happening in the invasion phase. So all of this is happening in the black box, and as said, we have some process that is getting unraveled, and as much as we learn, I think that our patients will benefit the most. So let's start by the endometrial microbiome. Well, let me repeat again the same concept. Basically, we are driving by population average. This is what generalized medicine is, which is what is better, to transfer embryos at day 2 or at day 3. What is better, to transfer embryos with 4 days or with 5 days of progesterone? No, what is better is to see the patient that you have in front of you with a specific biology, that then you will adjust, you will tailor your treatment according to the patient. And this is personalized medicine based on each individual person biology. And if we know this individual person biology, that's why I refer to personalization, we can use molecular objective tests that will have a biological plausibility that will be able to improve the treatment in this particular patient. That's the concept. So going to the first step, the endometrial microbiome. Let me just introduce the human microbiome world. As you know, we are living in a world, we are not only surrounded by microbes, but microbes, they are living on us, and they are helping us to produce very important functions. And in fact, the majority of these microbes, they are living in our skin, in our guts, in our digestive tract. Basically, if you weigh about 80 kilograms, 1 kilogram of your body weight is because of the bugs that they are living on. So this is an important element in our life. We have 10 times more bacteria than cells, and this is well known. And the progress that has been achieved now is because of technological progress. Because until now, only we can identify those bacteria because we culture them. If you culture something, you enrich them, so you have not have an unbiased overlook of what is there. Between 20 and 60% of the bacteria are cultured. Nowadays, we work with next generation sequences that allow us to investigate 16 ribosomal RNA of the bacteria, that with these hypervariable regions, we can get the identity of these guys without culture them, without anything. That's why the microbiome field has exploded, because for the first time, we are able to see them unbiased. So this human microbiome project has started more than 10 years ago, was promoted by NIH, and we learned what are the microbes that they are living on us, as mentioned, and the urogenital tract, about 10% of them, they are the one that they are there, mainly in the vagina, because this was the known at that time. So this is our contribution to the total body microbiome. But our question from about 7 years ago was, okay, we know about the vaginal microbiome, we know about the impact of the vaginal microbiome, not only in pregnancy, but also in obstetric outcome. But is there an endometrial microbiota? That was our main question. And the only data prior to the era of next generation sequencing was that in many papers, they tried to picture what is in the tip of the catheter and trying to relate them whether there was pregnancy or not. And all of this has the same conclusion. If you can grow bacterial pathogens from the EC, from the embryo transfer catheter, there is a decrease in implantation and there is an increase in discharge rate. This was quite obvious. So it was with this century, it was about 2000, that this technology was available. So we work, Immaculada Moreno is the principal investigator of this project. We work on the possibility to understand this. And what we did is to distinguish what is the results, the clinical results in patients. This was our first pilot study with few patients, but even so, we get shocked. That's why we follow this line of research, by which patients with what we call lactobacillus dominant microbiome, everything was okay. More than 90% of lactobacillus, that as you will see they are the good guys. Those are these blue guys that you have here. When you have this population, the only difference in these two populations is that when we analyze the microbiome, this was lactobacillus dominant, this was non-lactobacillus dominant. So we saw a huge difference in pregnancy rate, in miscarriage rate, 16 versus 60, and live birth rate, 58 to 6%. So, but even if it was a pilot, we got impressed from that. So we continued the research. So you can see this now in colors here. And we have next year, this was 2016, 17, a Chinese group published the fact that not only there is an endometrial microbiome, but there is a microbiota continuum through the reproductive tract, as you can see here, from the vagina, mostly lactobacillus, the uterus, but also the tubes, the ovary, and the peritoneum. So there is a continuum there, so it exists. Also, the uterus is a low biomass microbiome. So it's not like the gut. You have the percentage of bacteria per square feet. They are, of course, much lower, so you have to be very careful of that. And since then, there has been a number of published evidence, not only from our group, but also from other groups, including Richard Scott's group in the United States. And I just want to emphasize one, which was very interesting for us. This was a patient of mine that I was able to identify the microbiome in this particular patient after she got a miscarriage after transferring 2 euploid embryos. And then I just got the microbiome for a 4-week pregnancy of the same patient. Of course, I was not aware that she was pregnant. So I obtained endometrial fluid from her at the time that I have the checking. She has 1 day delay of her menses. And 1 week later, I find out that she was 4-week pregnant at that moment. Of course, the patient gave us the permission, she delivers, and the baby was fine. And that's why we report this as a special report in the American Journal of Obstetrics and Gynecology. Let me show you, this is just a 1-minute video that you can find in American Journal
in the human body can be found in the female reproductive system.
So what I have to go back?
The audience online can hear it, sorry, but not here.
Oh,
Maybe you can comment.
sorry.
Sorry.
So you're punished to be here, you're punished to have it. Okay.
Around 9%
Well, as mentioned, this is
can be found in the
basically
female reproductive system,
the
where bacteria
study that we did
facilitate
using
or hamper
16S
human
RNA.
Using
From
16S
this patient that she was a young patient,
we described the
26 years
human
old. She
endometrial
got a
microbiota
huge
in a
number of
patient who had
blastocysts,
an 8-week spontaneous
like 8
clinical miscarriage with
blastocysts,
euploid
euploids,
embryos, and for the first time during a successful
first
pregnancy
embryo
in
transfer,
which the
as mentioned,
endometrial fluid
and
was
she
sampled
got
at
a
4
miscarriage.
weeks of gestation.
Endpoint is that what we learned is
Lactobacillus
that
inerms
lactobacillus
is the most
was the
prevalent
main
microbe
population
found
in a
in
4-week
early
pregnancy with the embryo
associating
there,
its
and this
presence
was related
with
with the
defense
specific
mechanisms and
functions
basal
that you have
functions.
here, you can see in the paper.
The microbial
Whereas in the
profile prior to the
miscarriage from
spontaneous
euploid
abortion
embryos in
had
the very
higher
same
bacterial
patient, we
diversity and
found a
lower
diverse
lactobacillus
population in which
abundance, showing an
Streptococcus and
unstable
Barilla was
functional
there,
pattern
and the functions
characterized
were
by
also different.
transphosis and
So
insertion
this was
elements.
something that make us to
We
understand
found
what happens
taxonomic
at
and
that time
functional
in
differences
terms of the
between the
endometrial
microbiota
microbiome
and
with an
endometrial
initial
fluid collected
implantation
during
site
early
there, which I
successful
think that in this
pregnancy and
case
prior
it will
to
be
a
the
spontaneous
optimal
abortion in
microbiome
the same
because it
patient.
shows that it works for
Further
that.
investigation
So
of
that's
microbial
basically the
female
report of that with the
reproductive
specific
tract may come
functions
to
and the
understand
paper
the
you
role
can
of
see it
bacteria.
published. And the thing is, we continue with the results of our pilot study, and we covered this with a multi-center prospective study in which you can see here what was the features of the prior study and what are the features of this prospective multi-center study. Now we involve 13 centers from 8 countries, 3 continents, 342 patients. We took their endometrial fluid and biopsy samples, the system that we use to analyze evolved from the initial pyrosequencing to the ion-16S. We have more hypervariable regions here. We have more controls. We identify now the sample that they are low biomass, and we use some new programs for the contamination, for the background noise, and also for the interpretation of the taxa. So this is how we evolved in this prospective study. And basically what we asked the doctors is that in the patient, they have just to obtain in the prior cycle, transfer the embryo, and endometrial biopsy and endometrial fluid. Send it to us, go ahead with the same process, and then we relate the microbiome results with the clinical results. And basically we distinguish between good results, which is live birth, and bad results or reproductive failure, which means no pregnancy, clinical miscarriage, or biochemical miscarriage. And to make a long history short, basically what we saw here, that it was a clear association in the presence of this bacterial taxa when we have poor outcomes compared to live birth. So this was the signature of microorganisms related with reproductive failure, both in endometrial fluid and endometrial biopsy, whereas this is the signature present in good results in live birth, mainly lactobacillus. And whether this is lactobacillus iners crispatus, there are some controversial. However, in our study, we saw iners, although they think that this is crispatus. This is so. This is from the bench, basically trying to understand the existence of this microbiome. Now to relate this, to associate this with the poor or good outcome. And just very recently, we have this publication from a Japanese group in which they use actually the EMMA test, which is the test that we produce with this concept, which is 16S ribosomal RNA, and they act on that. They just treat those patients. So this is the chart that they use. They have these 200 patients, 64 of them. They refused to have the EMMA test, so they follow the line as they have it. And in 131, they did test. 64, they were normal. 44, they were ultra low biomass. So when you have biomass, you don't see lactobacillus, but it is because this is a biomass. So this is not pathology. This is just that you cannot see it, and this is not bad. This is okay. We know this now. But in these 30 cases, they were abnormal. And what they did is to treat this abnormal microbiome according to the microbe that they have. They give a specific treatment, and they find out that in the second biopsy, 75% of the patients, 23 out of 30, they were, among brackets, cured. So the pathogens were not anymore, so they move forward to transfer the embryos. And the results that they got is that after the first frozen embryo transfer, you can see here what is the results in the non-tested group, and you have here what happened in the tested group. So they found that when they treat those patients, clinical pregnancy, live birth increases at the very first frozen embryo transfer. And they also did a cumulative frozen embryo transfer after 2 cycles. And again, similar features were found. So somehow now they pass to the action. They have treated these patients that they found this biotic microbiome, and the results, they are getting better. So this is the whole history of the microbiome. And that's why I think that the conclusions could be that there is a pathogenic profile associated with reproductive failure. And this is quite clear. Lactobacillus are consistently enriched always with you have a live birth. These are the good guys. It doesn't mean that you can fix everything by adding lactobacillus, by adding probiotics. It means that you have to treat first, remove the bad guys, and once you do this, you can replenish the microbiome by adding lactobacillus, but not on the other way around. And finally, this were, although this is This has the limitations, but this is the first work, at least, that they do this personalized treatment using this 16S ribosomal RNA, and they show that they can improve IVF outcomes in patients with recurrent implantation failure. I forgot to mention that all of these 200 patients were those difficult patients with reproductive implantation, with recurrent implantation failure, which is quite important. And let me go to the next step, as I mentioned, which is the initial attachment of the embryo to the endometrial, to the luminal endometrium. This is possible because the luminal epithelium, it gets modified according to the hormones produced at this moment, mainly progesterone is the activation of the progesterone receptor, and it just push the production of the plasma membrane transformation, which means that now the cytoskeleton changes and this epithelium gets ready to get adhered by the embryo. This is the only moment when it happens. As you know, because you are working with embryos all the day long, the blastocyst, once gets his adhesion capability, is able to adhere to any place, including the transfer catheter, and in fact, including the plastic, including the microphone, whatever. In fact, you have to repeat the embryo transfer sometimes because it gets attached to the prostate, right? But the only place in the world that the embryo will not be able to adhere is in a non-receptive endometrium. That's why you have to find the right time, which is when the window is open. And you can check that out by not giving progesterone to your patient and see what happens. So that's the point here. So this is a very specific function that needs these requirements in order for an organism that is so sticky that is not able to get started in this situation. So, and we have been, the issue is how can we know that this patient is receptive? We have been trying to figure out this from the anatomical medicine, the Noyes criteria, using ultrasound, using electron microscopy, the binoculars, and but obviously nothing works because anatomical medicine has been already surpassed by the genetic medicine. And this is why we moved to the molecular medicine more than 20 years ago with other groups, trying to find out the specific transcriptomic signature that indicates at what time this window will be open. And this is what we did. And we came in 2011 with this signature, which composes 232 genes. That's with a different strength in this core. We have a strong core and then a core around that give us a prediction using an algorithm saying, now this patient is receptive. We did that in bulk endometrium, in the whole tissue. But then later on, we repeat that using single-cell RNA-seq, which means analyzing each cell, doing the transcriptomic, in this case, of more than 70,000 cells from the human endometrium. And by doing this, we repeat those results in terms of showing that there is you can see the change of color here. This marks specifically the opening of the window of implantation. So at this time, you have specific genes that they are turned on, and those are another ones that they are specifically turned off. So somehow it shows clearly that you can, something that you can see and you can act on that by predicting what is going to happen. This is different from the stromal compartment here, in which this deserialization process is more thorough, and you cannot identify when it started and when it finished. So the point is, by using this transcriptomic signature, we learned that the majority of patients, the window of implantation is after 5 days, 120 hours of progesterone activation, progesterone receptor activation, either by adding exogenous or endogenous progesterone. And this affects 70% of patients. But in 30% of cases, we learned that this window can be open after 6 or 7 days or 3 or 4 days. So, and the thing is, we do not know which patient will have the specific window, except if we test it, as it's logical. And then we came with the concept of the personalized embryo transfer as a treatment for recurrent implantation failure. This was our first point of endometrial origin, because it was the most difficult patient. So we came from that, and the concept was simple. Let's transfer the embryo, not only when the embryo is ready, which is what we do in regular embryo transfer, but when the window, the personalized window of implantation is also ready. So with this concept, again, going from the bench, now we went to the bedside. This was our first paper in recurrent implantation failure. And this was the very last paper published by a Chinese group now in the same topic, showing clinical pregnancy rate in the ERA group versus the frozen embryo transfer group. And what I can offer you, just to make a very long history short, because there has been more than 40 papers published about ERA with different results, with different specific indications in different parts of the world. This is the only meta-analysis that I have seen published so far, and they analyze specifically the clinical efficacy of ERA in patients with IVF-exit outcome. What they did is to divide between good prognosis patients and patients with recurrent implantation failure. And what they show in this paper, that after analyzing all the available data, recurrent implantation failure means 3 or more prior embryo transfer failed attempts, PET, guided by ERA is significantly increases the chances of pregnancy for non-receptive patients with recurrent implantation failure of endometrial origin. However, for good prognosis patients, even if the results shows from 39.5% with no ERA to 53 with ERA, this is not significant, and so there is no more data are needed in order to found this significance at this time. So this will be basically the reflection of this meta-analysis showing in numbers that this will be the live birth rate expectancy in recurrent implantation failure, that the window of implantation is identified and the transfer is being done personalized versus what happens with good prognosis patients, 53% with ERA, or with good prognosis patients with no ERA. So in conclusion, what I can get you from that, again, that there is a biological plausibility for the ERA, which is to understand what is the transcriptomic signature that indicates the opening of the window of implantation, which is personalized. And then if we transfer the embryos at that time, this is what is called the personalized embryo transfer. Our data that has been wide in these 10 years demonstrate that in 30% of our patients, the window of implantation is displaced. So we find the right moment and we will be able to synchronize the transfer better. And according to this first meta-analysis, ERA significantly increases the chances of pregnancy for non-receptive patients with risk of endometrial origin. And I just want to finish with 2 slides that I think that can illustrate what happens in our field, in general, in many other fields, but also in our field. Every scientific innovation that we produce follow the very same trends. And this is the example of PGTA, for instance. When PGTA was created, we talk about now 40 years ago, what happened was that first, there is what we call the panacea paradise. It is so wonderful, it is used for everything. Then comes the harmful hair. If you remember, in New England Journal, oh, you are killing the embryo, you are killing the mother, you are killing everybody. And then comes the consolidation period that with the time shows the reality of this technology. And if you see the HAMA paper now published last year, it demonstrates that 50% of the cycles in the USA are being done with PGTA. And you know that they have the better results that we have in the world. Far from the penetrance that we have in Europe, that is less than 10%. I learned that in your center, you are going to very high percentage of PGTA. So I congratulate you for being able to see this, because obviously if you transfer an employed embryo, it may work or not, but it will not work if you transfer an unemployed embryo. I think this is very straightforward. So similar feature is happening with the ERA test. The ERA test is now 12 years old, is just a teenager, and but we have the very same trend. First, we have this Panacea paralyze. It was useful for everything that you can imagine. Now, even we have an editorial saying that this is the end of the era. And now the consolidation period is coming, and now you have seen the first meta-analysis, and I hope that more data will come. But the basic point here is biological plausibility and personalization. And then let's see the clinical data will just indicate us what is the specific indication for each group of patients and for each patient in general and particular. And with that, I just want to mention, as I did before, that this is the foundation that I created. We are keeping working on the same field, on the same topics, but now in a non-profit situation. I have 50 people working with me. All of them, the majority, they were my PhDs since 30 years ago. Now they became PIs, and now we are in the second generation of PhD, the third generation of PhD students, basically. So this is the atmosphere that we are working for. And I would like to invite you to our first meeting that we will do in Paris this year, in which we will try to make a think tank of innovations in reproductive medicine. Not a regular meeting, but really a think tank meeting in which we will have always the triad, which is the clinical part, the basic part, and the visionary part. And how can we translate all of this knowledge from the bench to the bedside? Well, thank you very much, and I will be happy to address any questions.
Thank you, Carlos, for the very inspirational talk. You touched on some very important points in this talk that are very relevant for what we do in the clinic every day. So now I want to open the floor for any questions or comments from the audience. Don't be shy.
Hi. Oh, there's a microphone coming your way. Hi. Thank you so much for the leave to talk. I was wondering, some patients have indicated for antibiotics post egg retrieval to prevent infections. I was wondering if you think that kind of counter-indicates the endometrial microbiome you're talking about, and if they should be supplemented with probiotics during that time, or especially if they're a fresh transfer. What do you think about that?
Yeah, this is a very good point. You know, in fact, Fleming just anticipated the problem to give antibiotics to everybody without indication that this creates, basically destroys the microbiome and it creates problems. So to have antibiotics in general, after an oocyte retrieval, we always do because we fear that there will be an infection about that. Nowadays, there are many studies and many groups that they don't use antibiotics anymore after oocyte pickup. After embryo transfer, it doesn't make any sense to use antibiotics because what you are doing is to modify, and we don't know at what extent. So this is something that I will not suggest to do because it's like if you are treating antibiotics because you have a sunny day and you want to have antibiotics. So for the oocyte retrieval, to give in a specific treatments, it's not good. We have again to personalize, to know what we have, and to treat them properly. So this will be my recommendation.
I'll give you some time to think. Okay, while you think, I'll change to the online question. We have one. I understand the importance of the microbiome. Why don't we simply give probiotics to every patient rather than check the microbiome?
Right, this is the comment that I mentioned to you. Probiotic mean to give lactobacillus. So if you have Gardnerella, which you can see papers published, we published one paper in which I follow one patient for 3 years that has Gardnerella, and I treat her with everything that you can imagine, first, second, third line. I did the complex microbiological test, and at the end, the biofilm that the Gardnerella created prevent every treatment, and everything goes wrong. And honestly, I have to confess that I was unable to that this patient achieve a pregnancy. Probiotic is not the way to. If you give lactobacillus and still there is pathogen there, the probiotics will be useless. Probiotics can be used after you remove the specific infection, or if you have low biomass, it's okay to give probiotics. But again, it's the same issue with the antibiotics. Not generalize, please. Think in individual, think in persons, think in patients, not generalization. So generalization is quite easy to take, but it's not effective at all. So just see what is there, because if there is an infection, this infection should be treated in a personal manner.
Thank you, Maris. Oh, we have one in the back.
So expanding on your very well studied microbiome leads for healthy pregnancy, have you looked at the outcomes? Like, are they better weight range than better pregnancy, or anything like that? Quality that up?
You mean the way that the advert?
There's microbiome that's been in early child allergy, other things.
Right. No, this is a very good point. Now, in the study that we published in the journal Microbiome this year, this is prospective study with 300 patients. They were done in 12 different clinics. The thing is that, as you know, when the patient gets pregnant, she goes to a hospital to deliver. So we lose this data. We were focused on as you saw, reproductive success or failure. We were not went further than that. Because also the data are not in the centers that the patients were treated. So unfortunately, no, we don't have those. But I can tell you that the microbiome in the initial part of life is key. So we know that what the baby is having at that moment, because the baby doesn't have any microbiome until it gets to this world. And there was studies about whether C-section or vaginal delivery will make a difference, and even they got a technique which was to really go to the baby and give them the vaginal microbiome of the mother, just to those with C-section, just to assure that they saw so this was useless. So at the end, so this type of things is what we have to. This was not worth it. What is true is that the baby start to deliver their own microbiome in the very first week of pregnancy. But again, this has to do a lot with whether he has breastfeeding or he has just artificial milk or something. So it has many things to do with the behavior later on, not with whether this was a C-section or the vaginal delivery. So, but I don't want to go further on it.
Okay, so while we have another one, let's go.
Fabulous Emma data. Could you comment on the relative utility of Emma and Alice? For what portion of patients would Alice be useful or effective?
Right. Alice. We only have one, I'm sorry. Yeah, thank you. This is a very good question. Alice is, as you can see, every test that we do has a female name. This is of me. So it was Emma, Alice, and Embrace that we have. This is the non-invasive. Let me tell you the history. I wanted that it was named after Carmen Rubio, who was the principal investigator of that. But she convinced me that we have to use it. So, but it's supposed to be Carmen. No, Alice is because it's just the 7 pathogens that produce chronic endometritis. So Emma is the full microbiome, 16S metagenomics, microbiome, all of them. And Alice is just specifically devoted for the 7 pathogens with chronic endometritis. So this is something like analysis with chronic endometritis, Alice. So that's the difference. Because what we learned is that the chronic endometritis is also part of an altered endometrial microbiome, specifically affecting these 7 pathogens that produce the effect that we know nowadays as chronic endometritis.
Thank you for the question. Keep them coming. So let's take turns. I'll ask one of the ones we have online. So what is the benefit of doing an ERA if we can just add another day of progesterone for those that are pre-receptive, for example?
What is the benefit? Well, exactly this, that you know exactly where, when to add this 1 day or 2 days of progesterone, because you will have the window of implantation open, because if you miss the window, results will be bad. So it's about synchronization of the patient that you are treating. You know the embryos, you know that you have slower embryos and faster embryos. Same is with the endometrium. So some patients, they have a slow endometrium or faster endometrium. So to know exactly where is the window of implantation and to be able to really add more days. And in fact, you can see all of these clinical cases of patients with 7, 8 implantation failure. Once we detect that the window was at day 7, they transfer the embryos at day 7 and they become pregnant. So I think that this is the real benefit. It's as, you know, let's make the example in oncology that you need to have with every diagnosis and a specific gene or mutation that may change the whole profile of the treatment, making the difference between life and death. So in this case, this is the accompanying diagnosis that will let you know what the personalization means in terms of window of implantation for this particular patient.
Yes, like you mentioned about the precision medicine.
Sorry.
Okay. Do we have one? I'm sorry.
Thank you.
Just on that, had an idea as to, I was wondering if you had any evidence in previous studies of a person's window of receptivity changing throughout their life, throughout their treatment. Is there any guarantee that if you do an ERA, find someone's window, that window may shift during their life with the years of treatment?
This is an excellent question. We have been working on that a lot. First, the window of implantation in terms of if you are maintaining your life as usual, the window will be the same all your life. This is a transcriptomic signature that depends in this organ of hormonal regulation. So in the very first studies, we have donors being followed for 3 years. And when we analyze this patient at the same day with the same type of cycle, mean HRT or natural cycle, results were the same. And also we have now the second generation of babies born with ERA in the mothers. And when they come, you do not have to repeat the ERA test because you can just be consistent and transfer again at the same time. Be careful that the treatment will be the same. That's why we advocate to use HRT treatment because it is very consistent. Once you give estradiol, you give progesterone, and you know that it will be always the same. A natural cycle will be more tricky, even if the patient is older, will be even trickier because things may change. The only reasons that your window of implantation will change, there are basically two. The first is the BMI, and this has been published, we have published this already. So if there is a 10% change in your BMI, it may change the window because there is a connection between receptivity genes and metabolism. So one is the obesity or just the opposite. Even if this happens, it will be justified to see at what extent this is different. And for instance, endometriosis, we do not see any difference in the window of implantation. And we have done this, so this is not related, at least to this change. So the only reason was exactly what I mentioned, change in body in BMI in order to check again the window of implantation.
Thank you for the question. Okay, let's jump for another one from one we're coming remotely. So how to know that the EMA results, the microbiome test results, are bacteria that are alive? Because I guess what we identify in the samples is the bacterial DNA. So is that DNA from bacteria that is already, the cell is burst or not alive anymore? How can we know that the results are alive bacteria?
This is a very good question. Exactly. I mean, what is DNA? Is bacterial DNA. So it means that the bacteria either is there or has been there before. You can find this. The only way to do that is the metatranscriptomics, to see the RNA of those bacteria that they are living. So what we have done to address this specific question is from the paper of the microbiome that I mentioned to you. We have a collaboration with Ferring, which is interested in that. And we have done the metatranscriptomics of all of these samples now. And this is something that is going to be published very soon, showing that indeed, in those patients that we found this ribosomal RNA, they were the RNA was there. So it is not only the DNA, but also the RNA was active. But this is metatranscriptomics that as the person that asked the question find that this was our main concern. And the low biomass came also with the concept of how is possible. But this is about a very low abundance of this bacteria. And if you have less bacteria, the RNA will be less. But yes, we can just say today that really DNA or this analysis confronts with function, activity of the bacteria, not only with the person.
Very interesting. Anyone here? I'll just skip another one from the audience who's watching us remotely. So finally, what are the indications for endometrial receptivity testing? Which patient will benefit? Not everyone anymore.
Well, I think the indications you have to find that out with your own patients. But what I can see from evidence-based medicine in what I have presented you, I think that it's still valid for patients with recurrent implantation failure of endometrial origin. Obviously, if it's a patient with 43 years old, you have to look first for the embryo. That's quite obvious. But if not, you have the situation. This will be valid for the most difficult patients that we have. However, what we have done is to couple both the ERA and the metagenomics within the same analysis. So in the same analysis, once we obtain the endometrial biopsy or the endometrial fluid, we can analyze both the presence of the microbiome and where is the window of implantation. In this case, the indication, you have to find that out because now we are powering two different tests that will give you a scope of the situation of the endometrium. And I think that in the future, we will have more and more additions with real value to that. And it comes with the concept that the endometrium matters. It comes with the concept that, of course, the embryo is the star, no doubt about that. But if you have the best embryo that you have been working very hard and you have a Gardnerella, it will not implant. If you transfer with the wrong way of implantation, it will not implant. So the fact that you have the best embryo does not fix the whole process. So if you understand that the endometrial factor exists, now this test helps you to understand the window, but also the microbiome. And separate, if you use it separately, first one will be for recurrent implantation failure, and the second one, you have seen that patients with recurrent implantation failure improve results by being treated specifically And the other question is now, if we treat with antibiotics every patient, we don't need to do any test. No, it's not the case. So, so the point is that. So this is the indication in a separate fashion, but we can do both now at the same time.
I just have a question. If you had done any follow-up studies on like where they've already given birth, would their endometrium go back to being what it was before, or would pregnancy change the bacteria that are in there? Does that make sense? On like a second implant?
Let me understand your question. What do you mean is if pregnancy has made a difference?
Mm-hmm.
The difference for the microbiome can be made for many different issues.
If you're pregnant and you give birth, would you expect it to go back, the microbiome to go back, what it was like before?
Yes.
Change.
Yes, you do expect that, but if you have whatever infection, it will not go back. You have a postpartum fever, or if you have any issues, or you need to have a manual removal of the placenta, it will change things. So, but yeah, in physiologically, if you have a normal delivery, and the microbiome will go back, which is basically lactobacillus dominant, only lactobacillus there. This is the difference with the gut. The gut, the physiological microbiome is a diverse microbiome. However, in our case, should be only one, and it will recover after pregnancy. But it depends very much on many issues. For instance, the sexual hygiene, even the diet will change things. If you are a carrier, you know, there are so many factors that needs to be known when the patient wants to become pregnant, because this is where the action is. So the vaginal microbiome is fine. If you have a vaginitis, it's logical to look at that and treat them specifically. But now we are talking about a different organ, but also it's a neighbor organ, is not the same.
Yeah, I thought it was an interesting question because it's regarding the microbiome that you were focusing on. But there is some discussion about whether also the receptivity can change after you gave birth. Once, do you have any comments on that?
No, receptivity doesn't change when you give birth, because I mean we have the data from the patient that come from the second baby or the third baby. After trying, we have the embryos frozen and we transfer them back at the very same time, and it works.
And also, I know we touched this topic already, but regarding age, you said the transcriptome of the woman will be the same throughout her entire life. But I do remember seeing a few posters last ASHRAE that commented a bit how genetic expression could change, could vary a little bit as the patient gets older. What do you think about this study?
You know, I always think that because you know you have two organs which is the spectrum of longevity. You have the organ that died first, which is the ovary, at 40 years start to die, at 45 and 50 is death. But you have an immortal organ, which is the uterus. Immortal means that you can have a baby with your uterus wherever you want. And we have patients with 76 years old giving birth, even 80 years old giving birth. And considering our situation that the life expectancy is increasing, I mean, we may consider this for the future. There are countries like my country that we cannot treat patients older than 50. There are other countries with different policies. Well, the point here is that you have your organ, which is the uterus, which is immortal. What I have learned from the uterus is exactly that really it is not affected for biological functions through your lifetime. And in fact, that's the main reason. With organ donation, I transfer my patients at 50 and they become pregnant with a very good results. There are some recent data showing that as the endometrium ages, there will be some changes on that. So this is something that we have to be aware of that. And I just saw data which is not published that suggests that when you become older, you have a high rate of displaced window of implantation. But I don't know whether these were when the time goes by happens, or those patients that have this displaced window of implantation, and that's why they are trying to become pregnant now at 40s or 50s, so you never know. So my recollection prior to the actual data was that the endometrium does not age because the uterus does not age. However, we have to be vigilant from the data. These subtle changes may happen with the age. But yeah, you have an immortal organ, remember. This is amazing.
Agreed. Another one.
And the patients that you were looking at that did you see any specific trends in like lifestyle factors? Like, you know, we've had a discussion around age, but anything else that would essentially give us a clue as to who was going to be high risk and likely need the ERA earlier, so that we can avoid then going through repeated implantation, essentially see a shorter time.
Yeah, this is a very good question, but unfortunately, epidemiology, which is what you referred to, cannot really. I mean, you can suspect if you have bad habits, if you have bad hygiene, or many things. You can suggest that, but there is no epidemiologically. You cannot say this is going to have an altered microbiome.
Very dynamic environment. Okay, I have two questions here that I would like to ask, but someone here wants to ask anything first. Are you sure? But okay, so I will follow up with one of the questions I have here. Also, okay, so we're changing the topic a little bit because you mentioned Embrace before when we started the round of Q&A, and then there's a question that came about that test, which is Embrace is the non-invasive PGTA. So what are your views with respect to the non-invasive PGTA? Will it eventually replace PGTA, or should it be seen as a complement to PGTA?
Well, I know that non-PGTA is not very popular in this country for reasons that there are not specifically scientific reasons, but I mean, we developed this together. There was another Chinese group that also delivered this, and we published the data, and they published the data. Scientifically, we have seen this concept growing and also make a lot of sense, because in medicine we are moving to non-invasive techniques. From surgery to whatever you can think of, we are moving to non-invasive. And also the fact that when we biopsy embryos, we just obtain 3-4 cells, and we can get all this information from these 3-4 cells. Now we are jumping to have the whole DNA secreted by the ball of 100 cells, more or less. So altogether, I think that makes all the sense. There are many data already produced. We published 3 papers initially. The first paper, we learned that the concordance rate was only 30%, which we say in the paper, it was a human reproduction. No way this technology is not going to fly because we don't have this. In this very first paper, we learned that the cell-free DNA secreted equivalents to half of the genome of one cell, and there was no difference in employed or unemployed embryos. And so this was an exploratory, but we say no. However, we keep working on that. We improve our detection technologies. We modify the IVF part, because we need that the embryo will be in contact with the conditioned media from day 3 for 40 hours. And with all of this, then we publish another paper with 100 blastocysts with Laura Mienzi, Genera Rome, in which we just found a concordance that when we just analyze embryos at day 6 to 80%. And finally, we publish our prospective trial in many different centers with more than 1,000 embryos, in which we just reached this 70%. Now we are in 80% concordance. The study is finished, and there are many centers all over the world, at least when I left Igenomix, that they were working with us. is Carmen Rubio, who is the main driver of this project, and she does the validation first, prior that the groups will be able to send us the fluid in order to analyze this. So bottom line is, I think that this is going to be the future of the technology. Concordance doesn't mean that the PGTA, invasive PGTA, is perfect, because it's far from perfect. And non-invasive means that you do not touch the embryo. So I know that you are very good in your lab, but you must know that when you start this, there's a learning curve, and we have been going, all of us, from this learning curve, and embryos are losing because we are biopsying them. So the fact that we can go from a non-invasive and get a good perspective of the haploidy or aneuploidy of the embryo, I think that this is the future, and it's going to happen. It's going to happen. Also, in some places like here, you have this situation that it will take more time because you have now bad experience for different reasons. In other parts of the world, they are moving forward to that. And again, data are accumulated. We have a randomized clinical trial ongoing, comparing just morphology, embryoscopes versus just embrace, so see what happens. And we are also working on the basics on that. We're using embryo imaging with Nicolas Plansta. We try to understand how it is possible that the most important molecule that an organism has, which is DNA, gets secreted by you. Because the first reaction from our colleagues saying, no, this is apoptotic. This is our useless cell. But when we just find out that this is go back to back with everything that we can see in the normal karyotype, in the karyotype that we do, then things gets different. So we try to understand, and we have now the paper in processing, in reviewing Cell. This is the second round of review. So I hope that when it gets published, you will see the data that we gather, understanding how it is possible that the DNA is secreted by the embryo and it's produced, it just emerge from the nucleus and get out of the cell. So altogether, I think that this is going to be the future. It will take different times, it will take more proof, different places, but I'm pretty sure.
Thank you so much, Carlos. Since we are approaching our end of this session, does anyone here in the audience has a last question for Carlos? No one here. So we have a few questions that were left unanswered, which we can always forward to you later on at a later stage. And I just, before we finish, I would just like you to comment a bit. What do you think is the future of the technologies that we discussed in our presentation?
Well, I think that we should be driving by knowledge, and we should be driving by knowledge done using biological plausibility, and then moving from the bench to the bedside. What I have never agreed with the concept to use things random and do just clinical studies and see statistical significance only. And this is the way that you compare everything with everything, but then what? So this was the case of scratching. There is no biological plausibility for scratching. Also, there is we do not know what PRP is having. So patients are being treated with PRP because they are good for the hair loss or what. But when they introduce PRP, you don't know what they are there. So I think you have to know what you are doing. Any diagnostic or treatment, we should have the biological plausibility, and then we can go to clinical studies. But keeping in mind that we are trying to personalize. So if you do a general clinical study, things may work or not, but at the end, you have to know exactly what type of patients are you working. So I really foresee that science is now the best that we have seen in our history. We are able to do almost everything scientifically. So we should really take advantage of the actual situation in order to move our field forward. Because our field is not in the best moment now. We have a good room for improvement. Imagine that an airline only delivers 40% of the passengers to the final destiny. 100 go to the plane, only 40 arrive to Melbourne. So this airline will be out of business like that. So the only reason that we are going and growing is because we are working with the most important biological code that a mother has when she wants to have a baby. But we have to be better off because it's not fair that we cannot improve this, that can be improved. We are very inefficient as humans in terms of reproduction, but that's why we are using all of our system and we are just controlling the whole process in front of our eyes. And with that, I hope that we will also be able to improve in the black box, which is the periconceptional space.
Very nice. So one more time, thank you so much, Carla. Thank you everyone for bearing with us up until now. And for the ones who are present here in Melbourne, we will have a small coffee break now. And for all the others watching us online, I want to thank you for giving us a little bit of your time. And I hope everything that was said here is food for thought. Thank you very much.